Hey A&Pers, this is Dr. Jones checking in with A&P 1 Lab 1 Video 5. Okay, we're here with the cell model. This is the little one. I have the big one standing by so we can catch the rest of the structures. This is page 9 of the green booklet. So here is our cell model. This is a representation of the most common structures in a cell. There is no cell in your body that looks like this, but you know, it's got all the structures we need to talk about. So the outside edge of the cell is the cell membrane, and we'll talk about that in lecture in great detail. The inside, the middle here, is the nucleus. The nucleus is the powerhouse of the cell. It's, well, no, not the powerhouse. It's more the control center. It kind of tells everything what to do. So this whole egg is the nucleus. The egg shell is the nuclear envelope or nuclear membrane. And inside the little golf balls or hail are the nucleoli. Ooh, we haven't talked about Latin endings yet. Okay, so for Latin endings, and I'm not going to test you over these, but if you know them, it makes understanding where these terms are coming from simpler. So 1 nucleus, 2 nuclei. Okay. S for singular, that means 1. P for plural, that means more than 1. So same for here. This is a nucleolus, so the plural is nucleoli. Okay. And also you should know that the smaller the structure, the bigger the name. They tended to name the big things first, so there's a nucleus. And then as you get smaller, the short names are already taken, so the names get longer. Nucleus, nucleolus, nucleus, nucleolus. And inside the nucleus is the chromatin. Chromatin is the DNA in a non-dividing cell. So it's diffuse, it's uncoiled, and it is in the functional state. And we'll talk about that. The rough endoplasmic reticulum is here. It has the red bumps on it, red bumps. The smooth endoplasmic reticulum is the same stuff, but without the red bumps. The Golgi body is this red wonky bit in the corner. And we'll talk in lecture about what those do. The ribosomes are the red dots. So we have 2 types of ribosomes. We have the ribosomes that are stuck on the rough endoplasmic reticulum. They are stuck or attached, or it's also called affixed. So these are fixed ribosomes because they're attached to the rough endoplasmic reticulum. And these are free ribosomes because they are floating around in the cytosol, which is the liquid portion of the inside of the cell. So red dots on the RER. RER is not for credit on the lab test. Spell it out, rough endoplasmic reticulum. The red dots on the RER are fixed ribosomes, and the floating red dots are free ribosomes. So ribosomes is not full credit for either of those. The carrots here are mitochondria, and you can tell by this shape here, that's what tells you that it's a mitochondrion. And I've got a bigger mitochondrion over here to the side to show you what I mean. Last structure on this model though, these 2 Christmas candies, Twizzler nubs, are called Centrioles, and they are always at right angles to each other. And we didn't talk about what the cell parts do. Okay. This is a mitochondrion, but much larger. And so you see these folds here are what those are supposed to be showing you. So mitochondria will always be distinct because they have little folds in them. So parts of the mitochondria that you need to know. This outer layer is called the outer membrane. And then this inner layer is the inner membrane, outer membrane, inner membrane. Oh, that wasn't on screen. Okay, outer membrane, inner membrane. And then these folds in the inner membrane are called crista. Cristae for plural, crista for singular. We haven't covered that yet, have we? And then the stuff inside the inner membrane, this pink stuff on this model, is called matrix. And then for A&P 2, you'll need to know the space between the inner membrane and the outer membrane is the intermembranous space. But we'll get there. Okay. So let me move our giant mitochondrion. Oh, and a single mitochondrion, although I believe these are Greek endings, not Latin. And plural mitochondria. One mitochondrion, two mitochondria. Okay, the big cell. Okay, so this is all the same stuff as the little cell, but bigger. Let's see if I can move the thing on the tripod and I might have to move, okay. Okay, just a second. I can fix it. It's just a different doobie whopper. Okay, sort of better. Okay, going with it. So let's find all the same stuff again on this other model. So the edge of the cell is the cell membrane, and that keeps the good stuff in and the bad stuff out, decides what gets in and out, which is a term called permeability. This giant egg in the center is the nucleus. It's the control center. The egg shell is the nuclear envelope or nuclear membrane. For lecture, these holes in the nuclear envelope will be very important. Those are not lab terms, but they are lecture terms. These are called the nuclear pores. Inside the nucleus are the nucleoli, because we have two of them here. You can have one or two or lots. And then the red fuzzy stuff is the chromatin. Chroma means color, and it does stain nicely on slides. We also need to talk about, what are these with the red dots? Rough endoplasmic reticulum, and what specifically are the red dots called? Fixed ribosomes. Here we have two giant Christmas candies at 90-degree angles to each other, or Twizzler nubs, depending on how you see those. What are those called? Centrioles. I didn't mention it before, but centrioles are important for cell division. Cells that don't have centrioles can't divide. And we'll talk about cell division here in a second on the next video. What are the carrots? Those are mitochondria. And you can see the outer membrane, an inner membrane with folds called cristae, and inside the inner membrane is the matrix. And then the red dots floating around in the cytosol, free ribosomes. And the red wonky bit is the Golgi. And then the weird thing without the red dots is the smooth endoplasmic reticulum. And there are some other structures on here. There are probably peroxisomes and lysosomes, but that's not on our list, so we don't entirely care. But we do need to talk about these, because these aren't on the small model, they're only on this big one, and they are on the list. So these are secretory vesicles. Secretory because they secrete stuff, not secretary or administrative assistants. They're not taking notes or keeping track of your stuff for you. Secretory because they secrete stuff outside the cell. So let's talk about what these cell parts do. The ribosomes, both the free ribosomes and the fixed ribosomes, make proteins. So the free ribosomes make proteins for use inside the cell. As the proteins are made, they float off into the cytosol, which is the fluid portion of the cell, and they get used inside the cell. fixed ribosomes make proteins, but when they make the proteins, they go into these chambers of the rough endoplasmic reticulum. And then those products get packaged into a transport vesicle, which goes from the rough endoplasmic reticulum to the Golgi body. So the rough endoplasmic reticulum is the location of synthesis, and to synthesize something just means to make something. It's the synthesis of proteins for use outside the cell. The SER is also manufacturing, so this is protein manufacturing. In the RER, the SER manufactures carbs and lipids. So still manufacturing, but without the fixed ribosomes, can't make proteins, so they make carbs and lipids. So the RER manufactures proteins, synthesizes proteins, because the ribosomes make them and they catch them. And they go in transport vesicles from the RER to the Golgi. The Golgi is the Amazon warehouse of the cell, and it packages things for shipping and creates these secretory vesicles, which then secrete the stuff outside of the cell. Oh, and the nucleoli make the rRNA and assemble the ribosomes. The ribosomes are made of rRNA and proteins, so the nucleoli make the rRNA and assemble the rRNA with the proteins. Okay, we're past 10 minutes, so we're gonna call it good there. And I think we'll do the mitosis models next. So this has been A&P 1, Lab 1, Video 5, I think. Thank you, as always, for tuning in. The education must go on. Dr. J out.